A really important concept in in the field of biology is the surface area to volume ratio. And on this slide, I'd like to explain this to you. It's a concept and an idea which keeps on reappearing over and over and over again. The surface area to volume ratio limits the size of a cell. In other words, I try to answer the question now, why are cells so small? Why can you not see a big cell? Most of the cells are way too small to be seen with the unaided eye. And the answer why this is the case is a large cell would have a surface area which is too small compared to its volume. So, and this requires some understanding right now, because generally people think that if you have a larger object, a larger cell, then its surface area, its surface area is also bigger. Now that is correct, of course, and but not compared to its volume. So I'm going to illustrate this over here. I have two cubes, and these cubes are supposed to symbolize our cells. And cells are three-dimensional in structure, so they're not only flat, but they have a three-dimensional shape. And over here, this box over here has a side length of, let's say, 1 millimeter. Now that's of course way too big for a cell. It doesn't matter. Let's just use units that we're used to over here. So one from over here to over here, that is 1 millimeter, has a 1 millimeter length. The surface area now is 6 square millimeters. A cube has 6 sides. One side is 1 millimeter times 1 millimeter is 1 square millimeter. That's the area of one side multiplied by 6. You have 6 square millimeters. Now, if we double the side length, over here now this one side has a side length of 2 millimeters. So it's 2 times 2, which is 4, times 6 sides is 24 square millimeters. So, of course, when we make the object bigger, the cube bigger, the surface area, SA, also increases. I mean, that's kind of clear. So, but the question is now, what does this have to do with the surface area to volume ratio? Well, of course the surface area goes up, but so does its volume. So the volume of the small cube is 1 millimeter times 1 millimeter times 1 millimeter. And that is 1 cubic millimeter. The volume of the larger cube down here is 2 millimeters times 2 millimeters times 2 millimeters. 2 times 2 times 2 is 8 cubic millimeters. So while we have increased the surface area from over here to over here by 4, doubling the side length increases the surface area 4 times. The volume also increased from over here to over here. But by how much did it increase? 8 times. Because 8 is of course 8 times larger than 1. So even though both increase, you see that the volume increases significantly faster than its surface area. And this causes, from a certain size onwards, a problem for the cell. Because the surface area, which is the cell membrane, then, from a certain size onwards, will not be large enough to obtain all the nutrients that the cell needs. So it's a problem because the cell gets nutrients. The cell gets nutrients and oxygen over the surface area, that's the cell membrane, and removes waste over it. A cell that is too big would have a membrane which is too small for all of these events to occur. So that is the thing. Essentially, the cell has a problem growing further because it won't get all the oxygen, it won't get the nutrients, and so on. So what happens if the cell becomes too big? Then it has to divide to restore its surface area. That's the solution. Cell grows, becomes too big because the surface area to volume ratio drops, and therefore it has to divide. And after division, you have 2 small cells, each one again with a large surface area to volume ratio. And before I forget about that, the S to V ratio I mentioned over here. If you divide these two values, then you have you get a value of 6, and you can see that 6, of course, is larger over here. That this cell over here has a larger surface area to volume than this one over here. So this one has actually a twice as large surface area to volume than this one over here. Okay, so that was basically the concept, not only relevant for cells. The concept of large surface area reappears over and over and over again. Why do you have so many villi in your small intestine? To increase surface area. And why are there so many microvilli on the villi in the small intestine? To increase surface area even more. Why are there so many tiny blood capillaries? So that you have a large surface area over which to gas exchange carbon dioxide and oxygen. Why are there so many alveoli, small alveoli in your lungs? Large surface area for gas exchange, over and over and over again. So the concept of surface area is going to, I would say, follow you throughout the biology course.